In biogeography, parapatry is the relationship between organisms whose ranges do not significantly overlap but are immediately adjacent to each other. They only occur together in a narrow contact zone. This geographical distribution is opposed to sympatry in allopatry or parapatry. This distribution may cause speciation into sister species over time, a process called parapatric speciation. Parapatric speciation model. In the parapatric speciation model, the population of a species constitutes one or more biogeographically distinct subpopulations with a small, continuous overlap or minimal contact zone between populations. This minimal contact zone may be the result of unequal dispersal or distribution, incomplete geographical barriers, or divergent expressions of animal behavior, among other things. A parapatric population distribution may result in non-random mating and unequal gene flow, which can then produce an increase in the dimorphism between populations. Parapatric speciation is distinct from allopatric speciation, parapatric speciation, and sympatric speciation. In parapatric speciation, there is an intrinsic barrier of non-random mating and distinct selection pressures that create unequal gene flow. Parapatric speciation is the culmination of this unequal gene flow effect, in which genotypic dimorphism between populations results in speciation of the population and redefines the population as two or more sister species. Environmental gradients. Because of the continuous nature of parapatric population distribution, population niches will often overlap and produce a continuum in the species' ecological role across an environmental gradient. Whereas in allopatric or parapatric speciation, in which geographically isolated subpopulations may produce discretely separate niches, the reduced gene flow of parapatric speciation will often produce a cline in which a variation in evolutionary pressures causes a change to occur in allele frequencies within the gene pool between populations. This environmental gradient ultimately results in genetically distinct sister species. Ring species. Parapatric speciation can occur across a set of neighboring populations in which each can interbreed with closer populations, but end populations are too distantly related to interbreed. This population distribution of a species is referred to as a ring species. When an ancestral population extends its range around a geographical barrier and differentiates, reproductive isolation occurs between terminal populations. This results in technically continuous populations that undergo parapatric speciation. Fertility is negatively correlated with distance between populations, and terminal populations between ring species become distinct sister species. Examples. In the Martinique Senol, quantitative trait variation in dewlap and dorsum color pattern, body proportions and scalation demonstrated nascent parapatric speciation between adjacent sets of populations. Large quantitative trait divergences were observed with association differences in habitat rather than geographic allopatry. In the grass Anthoxanthem, mine contamination in an area has caused the species to undergo parapatric speciation by creating a selection pressure for tolerance of metal contaminants. This changes the flowering time of the grass and creates strong selection against interbreeding, causing many plants in the population to become self-pollinating. In the Gyrinophilis Tennessee cave salamander, qualitative timing of migration was used to infer the differences in gene flow between cave-dwelling and surface-dwelling continuous populations. Concentrated gene flow and mean migration time results inferred a heterogenetic distribution and continuous parapatric speciation between populations. In Ophidogymnosperms of North American, Bayesian inference of DNA molecular markers is used to estimate divergences of sister species. Ecological niche models are generated for parapatric and sympatric sister species. resulting in evidence for parapatric niche divergence for the sister species pairs E. californica and E. trifurcate. Parapatric speciation may be associated with differential landscape-dependent selection, even if there is a gene flow between two populations. Strong differential selection may impede assimilation and different species may eventually develop. Habitat differences may be more important in the development of reproductive isolation than the isolation time. Caucasian rock lizards Darevskia rudis, Valentini and D. Portchinsky all hybridize with each other in their hybrid zone. However, hybridization is stronger between D. Portchinsky and D. rudis, which separated earlier but live in similar habitats, than between D. Valentini and two other species, which separated later but live in climatically different habitats.